Mendelian Genetics. Mendel's model organism, the pea plant. Mendel chose pea plants as his model organism. There are many varieties with distinct inherited traits, such as color, height, and shape. Controlled matings can be achieved with pea plants. Each pea plant has both reproductive organs, the pollen-producing organs, stamens, and ovule-producing organs, carpels. Cross-pollination or fertilization between different plants can be achieved by dusting one plant with pollen from another. In order to precisely control his breedings, Mendel would cut the stamens off the plant to ensure no unwanted pollen or sperm would touch the carpels, and use a paintbrush to apply only specific pollen to the carpels. Mendel tracked 7 characteristics that varied in an either-or manner. There were 2 forms of each trait. These observable traits of an individual are known as the phenotype. So he tracked the seed form, the seed cotyledons, the flower color, the pod form, the pod color, the stem place, and the stem size. Here's a look at a typical experiment. Mendel started with 2 contrasting true breeding varieties. True breeding individuals will produce offspring identical to themselves when they self-pollinate or are crossed to another member of the same population. Hybridization is when 2 true breeding parents with different traits are bred together. The true breeding parents are the pea generation. You can see here the pea generation of green pods and yellow pods. One parent has green colored peas and the other has yellow colored peas. And when we breed them together or cross them, the hybrid first offspring of the pea generation are the F1 generation. In this case, all offspring have yellow colored peas. Monohybrid cross. Mendel crossed a true breeding pea plant having purple flowers with a true breeding pea plant having white flowers. This is a monohybrid cross because the parent plants differ in only one trait, their flower color. The observation was that all F1 plants had purple flowers. 2 F1 plants, so we take 2 F1 plants, cross them, and produce 929 F2 plants. The observation was that 725 of those had purple flowers, so about 3/4 of the plants have purple flowers, and 224, about 25%, have white flowers. Mendel concluded that the pea plants contained a heritable unit, later identified as a gene for flower color that was passed from generation to generation. He also concluded there were 2 versions, later identified as alleles, for the flower color gene. The white allele did not disappear in the purple F1 plants. Instead, the purple color allele was controlling the flower color gene in the F1 plants. And the F1 plants carried 2 alleles for the flower color gene, 1 purple and 1 white. Is this a pattern of inheritance? Mendel observed these same patterns of inheritance for the 6 other studied traits in pea plants. The F1 generation all had the same phenotype, which came from one of the parents. In the F2 generation, 3/4 of the population had the same phenotype as the F1 generation, and 1/4 had the phenotype of the other parent in the P generation. The experiments led to 4 principles. Mendel used words like heritable factors and elementin, as the work done linking chromosomes to heredity was not done until after his death. We'll use the modern terminology for clarity. Alleles are alternative forms of genes that account for variations in inherited characteristics. For example, there are 2 different versions of the gene for flower color in pea plants, one for purple and one for white. All organisms have two alleles. For each characteristic, an organism inherits two alleles, one from each parent. The two alleles may be the same or they may be different. An organism that has two identical alleles for a gene is homozygous for that gene. An organism that has two different alleles for a gene is heterozygous for that gene. Dominant and recessive allele. If the two alleles of a pair are different, heterozygous, the one that determines the phenotype is the dominant allele. The other allele that has no noticeable effect on the phenotype is the recessive allele. If we think back to the flower color on the pea plants, the purple that showed up in the F1 generation is the dominant allele, and white is the recessive allele. Mendel's laws. Although Mendel did not study chromosomes, his laws of inheritance are based on the movement of chromosomes during meiosis. Meiosis is the biological basis of Mendel's laws, the law of segregation, and the law of independent assortment. Law of segregation. A gamete, a sex cell, carries only one allele for each trait because allele pairs separate or segregate from each other during meiosis. The law can be applied to the separation of alleles for a trait or gene on homologous chromosomes as well as to traits that are on different chromosomes. If a plant carries an allele for a tall pea plant and an allele for a short pea plant, in other words, heterozygous for height, its gametes will carry either the tall allele or the short allele, not both. When sperm and egg unite at fertilization, each contributes its one allele, so the offspring will have two alleles, one from each parent. Alleles don't blend. Phenotypes that appear to result from blending are caused by multiple genes, as will be shown in the next unit. Law of independent assortment. Each gene segregates independently of each other. The evidence that Mendel came up with to prove this will be covered later. Chromosome theory of inheritance. Chromosomes were first observed in the late 1800s, but it wasn't until about 1900 that biologists began to understand what their functions were. The research starting in the early 1900s led to the chromosome theory of inheritance. In 1903, Walter Sutton and Theodore Boveri independently linked Mendel's rules with the details of meiosis to formulate the chromosome theory of inheritance. The theory states that meiosis causes the patterns of inheritance observed by Mendel in his pea plants, and hereditary factors called genes are located on chromosomes. Allele pairs. Remember from our study of mitosis and meiosis that each diploid cell has two sets of homologous chromosomes. It is on these homologous chromosomes that the allele pairs for each trait, Mendel's second hypothesis, are found. So in a pair of homologous chromosomes, one is inherited from the male parent and the other from the female parent. So we have the paternal homologue and the maternal homologue. A genetic locus is the location of a particular gene on a chromosome. So you have one allele of that gene from the paternal homologue and one from the maternal. At each genetic locus, an individual has two alleles, one on each homologous chromosome.